Good morning, everyone. Myself, Kritika, Tutor Department of Physiology from Sri Lakshmi Narayana Institute of Medical Science and Puducherry. So, which is affiliated under the Bharat Institute of Higher Education and Research. So, myself, my topic is about neurotransmitter. So, what are the things we are going to discuss in this session? So, one first one is a definition of a neurotransmitter. Second one, criteria of a neurotransmitter and classification of a neurotransmitter. So in classification, it has acetylcholine, amino acids, peptides, and biogenic amines. So first, the definition, it is a chemical substance that acts as a mediator for a transmission of nerve impulse from one neuron to another neuron through a synapse. So which is like a chemical substance liberated at the nerve endings that helps to transfer the message in the form of a nerve impulse from the presynaptic neuron to postsynaptic neuron. So the neurotransmitter substances are also released from the neuron supplying a muscle or a gland. So some of the chemicals secreted from neurons, it enters the blood to circulate as a hormones. The chemicals are called the neurohormones. So for example, we can say the ADH, GnRH, and norepinephrine, etc. And second, and there are some criteria for a neurotransmitter. So the chemical must be synthesized in the neuron concerned, or each second it should be stored in the presynaptic endings and should be released at the synapse. And fourth one should have a specific receptors on the postsynaptic membrane to target an organ. And fifth one should be disposed by a suitable mechanism as soon as its action is over. So these are some criteria for a neurotransmitter. And third part, classification of a neurotransmitter. So this is a summary like a classification of neurotransmitters. So the neurotransmitter, it is divided into four types: amino acids, peptides, biogenic amines, and acetylcholine. So the most secreted from the neurotransmitter is the acetylcholine. So first we will see the amino acids. Amino acids are sub-classified into four: aspartic acid, which is an excitatory neurotransmitter; and glycine, it is an inhibitory neurotransmitter; and glutamic acid is an excitatory neurotransmitter; and GABA, it is an inhibitory neurotransmitter. So aspartic acid and glutamic acid, this both are excitatory neurons. So which is terminated from stimulation by chloride independent membrane transport system reabsorption across the presynaptic membrane. And second one, peptides. So peptides neurotransmitters are vasopressin, somatostatin, and neurotensin. So these three specialized functions on the hypothalamus and also acts as a cofactors elsewhere in the brain. And third one, biogenic amines. So biogenic amine neurotransmitters are norepinephrine, dopamine, serotonin, catecholamines, and indolamines. So norepinephrine and dopamine, this secretes a catecholamine. These both are catecholamines, and serotonin is different. It is an indolamine. So for this one, the main function is a specialized modulating the functions. And an acetylcholine, it secretes excitability. Sorry, it is the main functions of acetylcholine is as excitability from acetyl-CoA and choline. So first one, acetylcholine. So acetylcholine, it is found in the neurotransmitters in many parts of a neurosystem. So as I given below, there's all autonomic preganglionic neurons, postganglionic sympathetic neurons, postganglionic sympathetic neurons supplying as sweat glands and also in the blood vessels of a skeletal muscle. And the axis of a motor neurons arising from a spinal cord, so which is mainly from a neuromuscular junction. And many parts of a brain, including bed cells and also the motor cortex and also the basal ganglia and amacrine cells in the retina. So they in this area, acetylcholine is distributed also. So how this uh, acetylcholine is synthesized? So it is synthesized in the presynaptic ending. So from choline, plus acetylcholine. With the help of a choline acetyltransferase, it forms the acetylcholine. Acetylcholine plus OA. So ACH mainly it is stored in the clear vesicles in the presynaptic endings, and also it is released by the exocytosis initiated by the calcium nerve terminals. So the acetylcholine, whenever it is released into the synaptic cleft, it binds with the cholinergic receptors, which is located in the postsynaptic brain. So, so what is the receptor which is present in the postsynaptic brain? It is cholinergic receptors of two types. So one is the nicotinic and the muscarinic. So first we'll see the nicotinic receptors. So nicotinic receptors, it behave like a ligand-gated ion channels, but with some differences with the muscarinic acid action. So the receptors are motor enclave but an ganglion. It can be blocked by the bungarotoxin, whereas mecalamine blocks receptors at the ganglia but not at the neuromuscular junction. So this nicotinic receptors are the members of a superfamily of ligand-gated ion channels that induces the GABA A and glycine and few glutamate receptors. So each nicotinic receptors, it is made up of 5 subunits like alpha, beta, gamma, and delta, that forms a central channel which permits only the calcium and other ions to pass through when the receptor is activated. So these are 16 known subunits for the 5 units of the nicotinic receptors. While moving to the muscarinic receptors, so it is mostly found in the brain, involuntary muscles and glands, etc. So there are 5 subtypes of muscarinic receptors such as M1, M2, M3, M4, and M5. So through these are the specific blockers are available for each receptor subtypes, atrophy blocks all. So the receptors are serpentine membrane proteins coupled to G proteins, calcium channel, etc. for their intercellular activities. So these are the summarized difference between the muscarinic versus the nicotinic actions of an ACH. So this is a site of action. So muscarinic action, it is postsynaptic, it acts on the postsynaptic membranes in a cardiac muscle or glandular muscle. For a nicotinic action, all autonomic ganglia and the neuromuscular junction in the skeletal muscle. So characteristics of action, it is about resemble those like a mushroom poison muscarine, and also it is very slow on the onset and the actions are prolonged in. So for nicotinic action, it actually resembles the drug nicotine. Actions are quick in onset, whereas muscarinic acid is slow. So next, actions are of brief duration. And actions are antagonized by the atrophy, which is when it combines with the ACH receptors at the site of a muscarinic action. So for nicotinic action, is hexamethylene at autonomic ganglia and tubular brain at skeletal muscle. So next, catecholamines. So these catecholamines are the epinephrine, norepinephrine, and dopamine. Epinephrine is a adrenaline, norepinephrine is a noradrenaline, and it's a dopamine. So mostly it is found in the brain, spinal cord, and peripheral nerves. So the norepinephrine is mainly secreted by the postganglionic synaptic endings, and epinephrine is also secreted by the adrenal medulla. So these catecholamines are mainly stored in the dense core granules in the presynaptic terminals or released by the exocytosis when they bind to their respective receptors in the postsynaptic membrane. So the epinephrine and norepinephrine binds to the adrenergic receptors are all which is serpentine membrane proteins These adrenergic receptors are divided into two main types: alpha adrenergic receptors, beta adrenergic receptors. So, first alpha adrenergic receptor, it's subclassified into alpha 1 and alpha 2. Beta adrenergic receptor divided into three: beta 1, beta 2, and beta 3. And these receptors, which is mostly present in almost all the tissues of the body, the enzymes monoamine oxidase (MAO) and catechol-methyl transferase, which it is metabolized the epinephrine or norepinephrine respectively. For epinephrine, the enzyme monoamine oxidase; for norepinephrine, the catechol-methyl transferase. So next one, the dopamine, which is mainly secreted by the dopaminergic neurons, are bind to the dopamine receptors and the postsynaptic membrane, and mostly in the synapses in the brain. So microstrial dopaminergic neurons in the basal ganglia are important in the normal motor functions. So whereas the five types of dopamine receptors have been described, such as D1, D2, D3, and D4 and D5. All these dopaminergic receptors are membrane proteins, and many of them have the subtypes. So it is observed that the number of D2 and D4 receptors increases eosinophenia, as the use of a D2 receptor blocker is very helpful in the treatment of a schizophrenia, and D2 receptor blocker when it also binds with the D4 receptors. So next we'll see the histamine. So histamine, it is also a neurotransmitter as a well circulating hormone. This histaminergic neurons are present mainly in the hypothalamus and limbic system. So the histamine, it has three receptors, one is a H1, H2, and H3. So this control the behavioral function and also the important peripheral actions like the smooth muscle contraction, secretion of HCl in the stomach, increased by capillary permeability, and arteriolar dilatation, etc. So like smooth muscle contraction, it leads to the bronchospasm. And next, serotonin. Serotonin, it is also the neurotransmitter. It is a amine chloride produced from the amino acid tryptophan by the hydrolyzation and it carboxylation. So it is also called as a 5-HT. So HT means 5-hydroxy tryptamine is a transmitter. So it is a it acts through its seven receptors named as 5-HT1, 5-HT2, 5-HT3, 5-HT4, 5-HT5, 5-HT6, and 5-HT7. So this 5-HT1 is divided into 5-HT1A, 5-HT1B, 5-HT1C, 5-HT1D, 5-HT1E, and 5-HT1F. And also the 5-HT2, it is also subclassified into the 5-HT2A, 5-HT2B, and 5-HT2C. And also the 5-HT5 also subclassified into 5-HTA, and 5-HT5B. So the most of the receptors acts on the activating adenyl cyclase and phospholipase C. So serotoninogenic transmission in the raphe spinal pathway. So where the nucleus raphe matters to spinal cord neurons, it is an important component of the endogenous electrical system. So this serotonin is a mood elevator and a lysergic acid, the diethyl amide, so called LSD, and hallucinogenic agent is a serotonin agonist, which acts through the 5-HT2 receptors. So the serotonin is also a circulating chemical secreted by the many cells, which is called endochromaffin cells, platelets, basophil, etc. And takes part in, and also it takes part in the physiological processes like contraction of a smooth muscle, uh, formation of platelet, blood, etc. So already I say the functions of a serotonin. So next, aspartic acid or aspartate is it is a chief excitatory neurotransmitter of a cortical pyramidal cells. It doesn't have many functions of aspartate. So next, gamma aminobutyric acid. So gamma aminobutyric acid is a inhibitory major inhibitory neurotransmitter in brain and retina. It is formed from a glutamate by the decarboxylation by the action of an enzyme called glutamate decarboxylase. So it is present in the body fluids as a beta aminobutyrate. It is metabolized mainly by the transamination reaction by the enzyme called GABA transaminase to succinate in the Krebs cycle. So the receptors, it has a three receptors called GABA A, GABA B, and GABA C. So GABA A and GABA B receptors are present in the brain, and GABA C is present only in the retina. So the GABA A and C receptors are the ion channels that have the five subunits surrounding core semi-core, similar to that of a nicotinic receptors. So this brain proteins that are activation lead to the increased chloride conductance. So it has a difference. Tranquilizer drug like a benzodiazepines uh, act on these receptors. So GABA B receptor as a metabotrophic receptor. So it is compared to the G proteins that are activation produce increased potassium conductance, and also it decreases the calcium influx and decreased adenyl cyclase activity. So as I say, GABA A, GABA B, and GABA C. So the main functions of GABA, it already said it is a major inhibiting neurotransmitter in the brain. So the action of GABA, such as when it is increased calcium conductance, it leads to the increased potassium conductance and decreased calcium influx, for all lead to the hyperpolarization of a neuron, causing generation of IPSP. Recently, it has been observed that a chronic level stimulation of a GABA receptor in brain is aided by the GABA present in the intestinal fluid, which controls the neuronal discharge billions of neurons in the brain by decreasing the signal to the noise ratio. And also, it is observed that GABA activity declines with age, which decreases the specificity, retinal neuronal response. So the benzoyl drugs that are also used in sedatives for muscle relaxant, anti-convulsions, and anti-anxiety medicine, which is also increasing the calcium conductance via the GABA A receptors. So when it binds with the alpha subunit of a GABA receptors, at a second of benzene receptors has been found in the peripheral tissues, and also is steroid secreting endocrine cells. These are called peripheral benzodiazepine receptors. So this PBR only it is involved in the steroid synthesis. And next, the next functions it is about the progesterone and the deoxycorticosterone. Whenever it is a highly induces sleep, and also acts as the anesthetics by acting through the GABA A receptors. And also the alcohol and the barbiturates also act partially by the increasing the calcium conductance through the calcium channels. So when the anesthetics, when it acts like a GABA receptor, so and also the alcohol participates in many volatile inhaled anesthetic. It increases the calcium conductors by acting on a GABA and also the glycine receptor. So some anesthetic it is acts by the inhibiting in MTA and AMPA receptors. So instead of acting on GABA receptor, the local anesthetic produces anesthesia by blocking sodium channels in the peripheral nerves. So next one, glycine. So glycine, it is a both inhibitory and excitatory neurotransmitter. So this glycine receptor is a pentamer having two subunits called alpha subunit, beta subunit. So the glycine receptor, it is a calcium channel that increases the chloride conductance and produces the IPSO. So hence where the glycine is a main inhibitory neurotransmitter. It causes direct postsynaptic inhibition in the brainstem and the spinal cord. So, and also it acts as an excitatory neurotransmitter in some parts of the brain where it influences the activity of NMDA receptors, which is bound to the glutamate. For example, in the dorsal horn of the spinal cord, the glycine binds with the NMDA receptor and facilitates the brain transmissions. So the neurons which causes the direct inhibition, which is in the three categories, the neurons secrete the glycine, neurons that secrete the GABA, and the neurons that secrete both the glycine and the GABA. So GlyT2, the glycine transporter is present in neurons secreting glycine and GAD, and the GABA transporter is present in the neurons secreting the GABA. So next we'll see the glutamate. So glutamate is a common excitatory neurotransmitter in CNS that causes about 75% of excitatory transmission in the brain. It is formed from the alpha ketoglutarate in the Krebs cycle, and also it accumulates in the synaptic vesicles by the transporters BNP1. So it is released from a neuroterminal. It is taken up by the astrocytes that is converted it first to the glutamine and it transfers the glutamine to nerve ending, which is converted to the glutamate. So the glutamate it is used for making lesion. So glutamate, when it is removed by the astrocytes from synapse, however, the glutamate plates in a higher concentration that acts on the cell bodies of a neuron. Intensely, it increases the calcium influx into the cell that causes cell death. So thus, glutamate in a higher concentration acts as a excitotoxic. So therefore, the glutamate is used to produce a excitotoxic lesion in the brain in the experimental animals. So this type of response it is seen in the ischemia, hypoxia, and the hypoglycemia and trauma. This is one of the mechanisms to damage the brain tissue that occurs in the stroke. So substance P and other tachykines like substance P is a major tachykinin. It is a polypeptide containing 11 amino acids. It is found in the many parts of the CNS, intestine, and peripheral nerves. So these are the other tachykines such as a neurokinin A, neurokinin B, and neuropeptide K, and the neuropeptide alpha. So the substance P it is secreted by the nerve endings, the first order neurons of a pathway in the spinal cord. So there are three types of neurokinin receptors: NK1, NK2, and NK3. NK3. So NK1 and NK2 receptors are the G protein coupled receptors. So next, the NK1 receptors for substance P. NK2 is the receptor for neurokinin K, and NK3 is a receptor for neurokinin P. The substance P activates the phospholipase C and increases the intracellular concentration of IP3 and DAG. So the functions of substance P, it causes the substance P, it causes a transmission of a pain in the dorsal horn spinal cord. The nigrostric pathway in basal ganglia, it is a co-transmitter with the dopamine and its concentration is proportionate to the concentration for dopamine. In hypothalamus, it causes low endocrine modulation. It mediates acts on reflex in the skin. It mediates a peristalsis in the intestine, and it suppresses a mode. Recently, the NK1 receptor antagonist has been used as an antidepressant. So next, nitric acid, it is a important neurotransmitter in the neuromuscular junction between the inhibiting motor fibers of intrinsic nerve pulses and the muscle fibers of the GI tract. It is also called the endothelin-derived relaxation. So it is formed from the arginine by the endosynthase. The endosynthase requires the NADPH, which is also called NADPH diphorus. And also it acts as a mediator for the dilated effect of acetylcholine in small arteries. The peculiarity of nitric oxide, it is that of neither produced by the neural cells or not stored in the vesicles. It is also produced by the non-neuronal cells like intelligent cells of blood vessels. In the brain, it acts as a signal for the postsynaptic neuron to communicate with the presynaptic neurons in mediating the long-term potential and long-term depressions. Purines and plumitins also are neurotransmitters in this group or adenosine, ATP, uridine, and adenosine metabolites. First, we'll see the adenosine. Adenosine, it is a general present in the central nervous system and potent vasodilator in many regional circulation. It has a 4 subtypes of receptors: A1, A2A, A2B, and A3. So all these receptors are for G-coupled protein. So this adenosine acting through A2A and A2B receptors increases the C-camp formation and acting through the A1 and A3 receptor, which decreases the CAMP formation. So the A1 receptor, which acts like antagonists, are used in the treatment of a stroke to decrease the glutamate release and prevent the it prevents the excitotoxic effects of a glutamate. And at last, we'll see the ATP. So ATP, it is nothing but energy, which produces energy. It has recently been emerged as a neurotransmitter in CNS. So it also causes a rapid synaptic responses and it causes a fast neurophysiological responses. So it acts to the P2X and P2Y receptors that are widely distributed throughout our body. So the P2X receptors is called reconjugated ion channel receptors, which it has a 7 subtypes from P to P2X1 to P2X7. So these are the 8 P2Y receptor subtypes. There are 8 types of P2Y receptor subtypes: P2Y1, P2Y2, P2Y4, P2Y6, P2Y11, P2Y12, P2Y13, and P2Y4. So this receptors in a dorsal horn of a spinal cord that indicates the role of an ATP in transmission of sensory information in the spinal cord. Thank you.